Today, we have Annabelle Singer. Annabelle did her undergraduate work at Wesleyan, graduate work for PhD at UCSF in the lab of Lauren Frank, and did a postdoc at MIT with Ed Boyden before she wandered south out of the cold weather back down to spend some time with us. So is now an assistant professor in BME, just joined this summer, correct, officially? So she'll be telling us about her work, very exciting stuff with these heavily instrumented experimental setups. Looking forward to hearing about it.
All right, thank you very much, and thank you, Chris, for organizing. Can everyone hear me? Okay, great. All right, so let's make sure this works here. There we go. All right, so decades of research has shown how spiking activity in the brain reflects our experiences or encodes sensory information. For instance, John O'Keefe won the Nobel Prize a few years ago for his discovery of place fields. He found by recording from neurons as animals ran around an environment that neurons in the hippocampus fire when the animal's in a particular location in space. And if you took many neurons together, you could actually get a representation of that entire space, a kind of place field map. Now, we know how to read a map like this one, but we don't really understand how to read a map from place fields. And that's really a key question. How do we understand these patterns of activity that guide behavior? We really want to understand how this neural activity is decoded, ultimately in the service of behavior. So today what I'm going to talk to you about is, first, I'm going to focus on these patterns of activity, and not just how they encode experiences, but how they can inform future behavior. And we do that by decoding neural activity computationally. And with that method, we're able to bridge the gap between these place fields and behavior. And then I'm going to examine how these patterns of activity fail in mouse models of Alzheimer's disease. And that we do by detecting and manipulating activity in Alzheimer's mice. All right, so first, so how did we get here today? Now, we've known about place fields for a long time, but how does that information help us decide where to go next? Or more importantly, how does it help a little boy like this figure out how to get back to that house he remembers from last year where he got the really good Halloween candy? Now, place fields report, if you think about it, they're really reporting where am I right now? I'm here, I'm here, I'm here. But that isn't really the information you need if you're trying to figure out where to go next. So what I'm going to show you and what we think is happening is that when an animal has to choose a path, there are patterns of activity in the hippocampus that reactivate the possible paths that the animal can choose, essentially foreseeing where the animal is going to go. And it does this based on its past experience. So it's similar to this little boy being able to foresee or choose between these paths by foreseeing where he went based on his past experience of last year. You should know that the hippocampus is essential for forming and storing memories of events and facts. It's also essential for place fields. I don't think this is a coincidence. I think this is the spatial map is the neural substrate of this, but I won't talk about that extensively today. All right, so there are different kinds of activity in the hippocampus. I already told you a little bit about place fields. This is a cartoon of what they look like with the animal's position on the x-axis and the firing rate on the y-axis. And what you can see is the animal runs along this linear track, cell 1, then 2, then 3, then 4, then 5 fires. When the animal reaches a reward here, they pause, and you see a very different pattern of activity. In the local field potential, we record ripples. Those are high-frequency events, and there are bursts of population activity at the same time. And what's really interesting is that during those bursts, we see the same sequences of activity, the same patterns that we see during behavior. So cell 1, then 2, then 3, then 4, then 5 will fire. Now, for a long time, place fields have been thought to encode a spatial map, but this reactivation has been a little bit more mysterious, and we thought it was really well suited to inform behavior because it can tell you about places that are not right here. It's not just here I am right now, but it can tell you about other places in the environment. So we thought it might actually be really good for informing future decisions. So we're going to ask, does reactivation of these neural codes inform decisions? And to do that, we're going to examine neural activity while animals are performing a decision-making task. So in this task shown here, the animal has to learn to alternate. So they would go from here to the center, to here, back to the center, etc. Right? And animals learn this task by trial and error. So this is showing you training sessions on the x-axis and the proportion correct on the y-axis. You can see over training sessions, the animals get better and better at the task. And we know that rapidly learning this task is hippocampally dependent. So to examine the role of reactivation in decision making, we're going to compare spiking activity preceding correct trials and incorrect trials. So when the animal has just come along this path, this is the future correct trial, this is a future incorrect trial. So we can compare that activity before the animal does that correct path or incorrect path. So we're focusing on this activity when the animal's right here. They're stopped, they're getting a reward, and that's when we see this ripple reactivation. And this is showing you what some of that activity really looks like. This is when the animal's running right here. So the red line indicates the animal's position through space. These are place fields on the y-axis, and the black lines are spikes. This is what's called a raster plot. And what you see is as the animal runs, you see spiking in the place fields. When the animal stops, you see these bursts of population activity. If you were to zoom in on them, you would see the precise sequences that I showed you before. And then you see the animal run again. And what I want you to notice here is the animal spends a significant amount of time stopped. This is not inconsequential when you think about the entire time of a single trial here. Right? And this activity has often been ignored. So we're really shedding new light on this kind of activity. All right, so comparing the correct and incorrect trials, first, if we look just at when the animal is just exposed to the task, they don't perform it very well. So this is less than 65% correct. We're not then at that point seeing a difference in ripple activity. And what we're measuring is coactivation probability in sharp wave ripples. So that's essentially how much two cells fire together during ripples. Then we look at periods when the animal's just getting the task, so 65 to 85% correct, or they've just gotten, for the first time, over 85% correct. Now we start to see a significant difference between correct and incorrect. And what we're actually seeing is a lack of ripple activity preceding the incorrect trials. And this occurs specifically during learning. We can then use that activity to figure out if we can predict the animal's performance based on the sharp wave ripple activity on a trial-by-trial basis. So to do that, we take half the trials, we train a linear regression model, and then we predict correct or incorrect response based on just the ripple activity in the other half of the trials. And we see something like this. So the gray line here is chance. The dashed line is what we see, our correct prediction for before the animal has learned the task, when they're less than 65% correct. And the solid line is when they're just learning the task. And what we see is that we can predict significantly more trials just based on the ripple activity during this early learning period. So we're finding that we can predict correct versus incorrect choices, specifically during learning from this ripple activity. But then we want to know, what does that ripple activity preceding correct trials represent? Is it representing the past path where the animal came from, the future path the animal is going to take, or potentially its possible paths? Or, of course, some combination thereof. To do this, we need a way to determine what spatial paths those ripples represent. So from the animal running through space over and over again over many trials, we get this place field code. And then we can use that to figure out during ripples, if we look at when the animal stopped and we see ripple activity like this, where would the animal be if the animal were moving during that ripple? So this is showing you one ripple with activity, and these are 15 different place cells on the y-axis, and this is time on the x-axis. And you can already see a kind of diagonal pattern. So what that's showing you is the order in which those cells fired. These cells on the y-axis were ordered based on how far they are from the animal's current position. So this cell is the farthest away over here. This cell is the closest over here, meaning its place field is far or close. So what this diagonal pattern means is that cells that are farther away fire early in the ripple, cells that are close fire late in the ripple. But to actually quantify that, we do a kind of decoding. And so what we do is we plot the decoded distance based from where the animal's current position is. So for each ripple, we divide it into 15 millisecond bins. This is showing you one of those bins. We take the spiking activity in that bin and we decode the animal's position if it were moving in space based on this place field code. And for that bin, we get the animal's most likely position would be about 120 centimeters. If we march through that ripple, we can do that for each 15 millisecond bin, and this is what happens. All right, and what I think you can see is that early in the ripple, this blue here and blue here, we're getting decoding farther away or higher numbers, versus late in the ripple, the reds and oranges, we're getting decoding closer to the animal's current position. So even though we're stopped, or the animal's stopped, we are getting a decoded or coherent path through space, and that's a path like this in this particular case. Now this is just one example, but we're going to use this approach to figure out what paths are reactivated when an animal has to make a decision. All right, so first we're going to ask a really simple question. If we decode those ripples, do we see paths more in line with an inbound movement through space or an outbound movement? And we're going to be looking only at correct trials in this case. What we find is that there's a higher proportion of ripples that represent outbound directions than inbound directions. And these are these two performance categories we talked about before, where the animal's really just getting the task. Now then, for the outbound direction, we want to know if the animal is, or if the ripple is decoding the future correct trial or the future incorrect trial. So if the animal just took this path, the future correct trial is going to the other arm. The future incorrect trial is going to back to the same arm. In this case, we don't find a significant difference between future correct and future incorrect paths. We do see a little bit of a trend for the future correct path, but overall we think that their possible paths are being reactivated during these ripples preceding correct trials. All right, so we're finding that cells are more coactive during ripples preceding correct trials, specifically during learning, and ripple activity preceding correct trials represents possible paths. And this is really a novel discovery linking place field codes to decision making. All right, so next we're going to focus on how does this activity go awry in mouse models of Alzheimer's disease. Now, there's a lot of bad news about Alzheimer's disease. I'm not going to tell you all about it today, but I'm going to focus on a few key points. One of them is that we aren't really making very good progress in terms of reducing the number of deaths due to the disease. This is showing you a plot of the change in the number of deaths between 2000 and 2010 for many different leading causes of disease in the US. And what you see is that we're making progress in things like HIV and stroke, but we're really not making progress in Alzheimer's disease. Part of the reason for that is that current therapies are terrible. And so we really need some better approaches and some new insights into this disease. One of the things you should know, you probably know that memory goes wrong in Alzheimer's disease, but you may not know that one of the earliest symptoms are deficits in spatial navigation. And one of the brain regions that's first affected is the hippocampus. And something else that I find really enlightening as a neuroscientist is that this progression of the disease is not a simple decrease. So if you're looking at memory function or other cognitive function as a function of the disease progression, you don't just see a line like this dark green line that goes sort of progressively down. It's not that simple. That is an average really of a vast amount of fluctuation around that line. So caregivers will say things like, this morning he seemed totally fine, but right now he couldn't find his way out of this room. Right? And what that suggests to me is that there's some kind of spontaneous shift from function to dysfunction. And that's most likely to happen, at least in my mind, I think, on the network level. It can't be explained by something like neural death. That happens late in the disease, more like here, but it's also not reversible. You don't get this spontaneous rescue and dysfunction. All right, so it's generally assumed that proteins accumulate in the disease, in particular amyloid beta, and that leads to a host of neurotoxic events, ultimately leading to failures in neural activity and deficits in learning and memory. But this level in particular has not been very well studied. I think part of that is because it's hard to study this, even in animal models. It's hard to look at this activity because you really need to look at it during behavior. But I also think it hasn't been studied because there really hasn't been that much we could do about it. So what? If you can't rescue it, why bother, right? But things are changing now. We now have tools to be able to study it and tools to be able to rescue this kind of activity. So one of the tools that we use is shown here. This is a mouse running through a virtual navigation environment, or virtual reality environment. And the animal's head fixed, and that makes it a lot easier for us to be able to record neural activity. This is showing you some example traces from a recording from the hippocampus, shown in red. And it also makes it a lot easier for us to manipulate that neural activity, in this case, using optogenetics. All right, so we're going to ask first, how does neural activity fail in a mouse model of Alzheimer's disease? And then what happens when we drive the activity that is lacking? I've already told you about sharp wave ripples and how we see differences between correct and incorrect trials. So they are a potential likely culprit for failed activity in the animal model. But another potential culprit is gamma oscillations. There's already work from humans showing that they're altered in Alzheimer's disease. They're also been shown to be altered in other mouse models of Alzheimer's. And gamma oscillations are oscillations that we detect in the local field potential. That's extracellular recordings that represent a lot of neurons at the same time. And we think that they really represent oscillating levels of inhibition. So cells are more likely to fire in particular phases of gamma, and that makes them more likely to fire together. That's important because cells that fire together are more likely to drive downstream neurons. They're more likely to drive plasticity. So things that fire together have a lot of significance. Gamma oscillations have also been shown to increase during learning and memory. Again, because we think that that's significant because neurons are firing together in short time windows that are required for plasticity. In the hippocampus, we observe gamma during both theta, when the animal's running, and sharp wave ripples, like I've shown you before. And something to note about those sharp wave ripples is that they're indicative of a kind of global brain state where the hippocampus is communicating with cortex. And we think gamma is probably part of the way that the brain regions get synchronized. All right, so we are recording from 3-month-old 5X FAD mice. This is a mouse model of Alzheimer's disease. It's pretty aggressive. It has 5 familial AD mutations. At 3 months old, we see some AD pathology, but mostly it's elevated amyloid. At that point, we don't have things like plaques, and we don't have observed learning and memory deficits yet. So it's somewhat early in the disease stage in this mouse model. Now, if we look at theta activity, this is showing you a power spectral density during theta, or during periods when the animal is running. And this is showing you frequency and normalized power. The power is normalized to the peak in each animal to deal with some differences in the recordings. And 5X FAD mice are in green, wild-type mice, the wild-type littermates are in black. And we don't really see a significant difference here. There may be differences that we aren't able to detect at this point, but there was really nothing that jumped out at us from that. So we also looked at sharp wave ripple activity. To do that, this is showing you a spectrogram with the time since the sharp wave ripple peak on the x-axis and the frequency on the y-axis. This red circle here is the typical ripple frequency, so around 150 hertz. So this is really showing you there is a ripple. But at the same time, we also see elevated lower frequencies, really anywhere from 0 to 50 hertz. And this is showing you in one Y. Animal. So in these examples, you can see we're really getting lower power animal. And if we look more specifically at what those instantaneous frequencies are, because this is a pretty broad range, what we find is that it's a unimodal distribution around 40 hertz. So that's often called slow gamma. And that was just 2 example animals. If we put all the animals together, this is showing you a cumulative distribution of the gamma power during those sharp wave ripples, that peak time. And we find that there's significantly lower gamma power in the 5x FAD animals than in the wild type animals. And what we really want to know is how does that affect firing? Like I said, gamma is important because it makes cells fire together in short time windows. So we measure that by looking at the fraction of spikes as a function of gamma phase. So this is gamma phase on the x-axis here, and this is showing you one example gamma cycle. It's 2 cycles. And 5x FAD mice, they are both phase modulated by gamma, but the modulation is weaker in the 5x FAD animals. And this is just quantifying that, where you take the depth of modulation, which is the difference between the peak and the trough here. 5x FAD mice. So gamma during sharp wave ripples and sharp wave ripple abundance, which I'm not showing you, but that's the frequency of sharp wave ripples, is reduced in 5x FAD mice early in the disease. All right, so since gamma is associated with learning and memory, and it's altered in AD, this is potentially significant. You could imagine it might be a way that you get these learning and memory deficits in the AD animal and potentially in the human disease. But what's really interesting here is that we're finding these deficits early in the disease before detected behavior deficits. So then what happens when we drive gamma? Now, you might expect that I'm going to ask a question about behavior right now based on this plot that I showed you at the beginning. But we're actually going to turn this around. Before we look at behavior, we decided that we really wanted to flip this on its head and ask, how does that change in neural activity affect protein accumulation? That seems like a little bit of a strange question, but it's not totally unprecedented. Other people have shown that amyloid beta, the protein that accumulates and is thought to initiate neurotoxicity in this disease, that is affected by neural activity. So if you increase neural activity by stimulating whiskers, you can increase amyloid beta. If you decrease neural activity by removing whiskers, you can decrease amyloid beta. So because activity can affect amyloid levels, found early in the disease in this animal model, we want to know, can gamma alter amyloid beta levels? All right, so we're going to do this by using optogenetic stimulation of parvalbumin interneurons. Others have shown that you can drive gamma oscillations by stimulating these neurons. And the optogenetic protein we're using is channel rhodopsin, which makes the neurons fire in response to blue light. So we're using a 5x FAD mouse that's been crossed with a PV Cree mouse. So that allows us to specifically infect PV Cree or PV interneurons with the channel rhodopsin. And our controls, we have different controls that I'll talk about, but one of the key ones we use is an EYFP only control. That means that the animal has been infected with a virus, but it only expresses EYFP, not channel rhodopsin. Some of the other controls we use are different kinds of stimulation. So for driving gamma, we stimulate at 40 hertz. For a control, we do a random stimulation, and we also stimulate at other frequencies. And in these experiments I'm going to tell you about right now, we're targeting CA1. All right, so this is showing you an example trace. This is the local field potential. This is the stimulation when the laser turns on. And if we look at the power spectrum, we see for no stimulation and random stimulation, we don't see really a change in gamma, but for 40 hertz stimulation, we see actually a pretty sharp increase in 40 hertz power. If we look at spiking activity, this is multi-unit activity. This is spikes after the start of the pulse. And we see that mostly there's an increase in spiking around 2 to 3 milliseconds after the pulse. That's probably in direct response to the pulse of light. Between 40 hertz stimulation and random stimulation. And let me explain here, the random stimulation is, there's a randomized interval between the light pulses, but it's on average 40 hertz. That randomized interval is selected from a distribution of that centered around 40 hertz. So we overall are getting a similar amount of optogenetic stimulation, meaning light pulses and whatever the neural response is to that, but we are not driving this 40 hertz power. And that's important because optogenetic stimulation has a lot of advantages, but it's also a really artificial manipulation of the system. So having a nice control like that is important. Yeah, go ahead.
Do those all identify interneurons?
No. So they're most likely interneurons. So it's a great question. For people who can't hear, Bilal asked if those are interneurons. So we're recording multi-unit activity. We're recording with a glass electrode, so we don't have photoelectric effects. But it's not a great system for isolating interneurons or even single units. So this effect is most likely due to interneurons, but I can't say for sure. Simply, the reason I say that is simply because we know that if you do isolate interneurons, you would get a plot that looks like this. All right. So we're driving gamma by driving PV interneurons at 40 hertz. And with random stimulation, we're getting the same amount of stimulation, but no gamma. So this is showing you the amyloid beta levels for amyloid beta 140 and amyloid beta 142. Those are key to the disease. Where there's no opsin of light, and the 40 hertz stimulation. What you see is there's a significant reduction in amyloid levels in both amyloid beta 140 and amyloid beta 142 with the 40 hertz stimulation. Now that was driving PV interneurons. If we instead infect alpha CAMK2 positive cells, which are putative excitatory cells, we don't see a change. So this is showing you, again, EYFP and 40 hertz in both the amyloid beta 140 and amyloid beta 142. Something I'd like to note is this is soluble amyloid beta. In these animals, there's elevated soluble amyloid beta at this age, but there is not things like plaques. All right, so driving PV interneurons at 40 hertz significantly reduces soluble. The CAMK2 does not change amyloid beta. And then other frequencies, we don't see a decrease as well. I'm not showing you those here. I can if people are interested. All right, so we're finding that driving gamma reduces this pathogenic protein, amyloid beta, and that's kind of surprising. So then the question is, well, how? We looked at different aspects. So amyloid betas are levels of amyloid beta production and amyloid beta clearance. And we looked also at production, which I'm not going to tell you about that today because of time. Instead, I'm going to focus on clearance. One of the key aspects of clearance is thought to be these particular cells called microglia. And they have many functions in the brain, but one of them is to clear out debris and pathogens. So you can kind of think of them as like garbage collectors of the brain. So we asked, is this 40 hertz stimulation affecting these microglia? So this is showing you histology using a marker called IBA1 with the EYFP control, 40 hertz stimulation, and random stimulation. So these are, in green, is microglia. And we count the number of microglia, look at the cell body diameter, and the process length. So what we're finding with the 40 hertz stimulation is that there's a significant increase in the number of microglia in the particular region that we look at. There is an increase in the cell body diameter, and there's a decrease in the process length. So you may not know, but microglia can move, so we think that they're being recruited to the area. And we think that they're changing their morphology. And in particular, this change where the cell body increases and the process length decreases has been associated with increasing phagocytosis or engulfment. So we wondered if that might be what's going on here. Now, to note that we don't think this effect is due to changes in IBA levels. That's the antibody that we're stating for. Because we also those levels. And we also don't, so far anyway, we haven't seen a change in inflammatory genes. And the reason I bring that up is because people have often associated microglia with inflammation. There may be some changes that we don't fully understand yet. All right, so we're finding that 40 hertz recruits more microglia to the area and alters their morphology. And this morphology that we're finding is associated with engulfment. Okay, so then we wanted to look at, well, is there more amyloid beta? And what we find is a significant also are positive for amyloid beta. So what we think is going on is there's an increase in this co-localization, and we think that that is actually reflecting increased engulfment of the amyloid. So driving gamma seems to reduce this pathogenic protein amyloid beta, and we think that it's doing that by recruiting microglia to engulf the amyloid. But you might be wondering, I just mentioned optogenetics has some drawbacks. One of them is that it's very invasive, and microglia do react to all sorts of invaders. So we wanted to know if this activation could be confounded by the invasive nature of optogenetics. We also came up with a way to non-invasively drive this gamma. It also has potential translational properties if it turns out that this is a beneficial effect. So this was actually at the suggestion of Emery Brown. He pointed out to us that you can flicker lights off and on to drive particular frequencies of activity in the visual cortex. So you can flicker lights at 40 hertz and get 40 hertz in the visual cortex. And in these animals, there is elevated amyloid beta in the visual cortex. It's not the most relevant brain region to the overall disease, but it's useful for us testing this hypothesis. So if we flicker the lights off and on, at 40 hertz. And this is showing you the fraction of spikes from, as a function of the light turning on and off. So light on is in yellow, light off is in black. And what you see is that the fraction of spikes increases and decreases with the light turning off and on. And this is for a random flicker, so we aligned it to the light turning on. And then since the flicker is random, there's some kind of random off and on in the interim afterwards. And we don't see that periodic increase. So we can flicker lights at 40 hertz to induce 40 hertz oscillations and firing in visual cortex. And since visual cortex has elevated amyloid beta in these mice, we can use this as a non-invasive method to test if the 40 hertz oscillations reduce amyloid. So this is showing you amyloid beta 140 and amyloid beta 142 levels in these mice after dark in black, light in yellow, 40 hertz flicker in red, or random flicker in blue. And what you can see is that there's a significant reduction in amyloid beta after 40 hertz flicker, but not in the other conditions. We also did some additional experiments here. One was 20 hertz, which is shown here, 80 hertz, which is shown here, and then 40 hertz along with picotoxin. So what you should note is that none of these have this reduction. The 40 hertz with picotoxin is, that's a GABA antagonist, so we're getting rid of GABA. We're significantly reducing GABA, and that gets rid of the effect. So we think this effect is GABA dependent. So 40 hertz light flicker reduced amyloid beta relative to dark or light only controls and compared to random light flicker. And the effect is GABA and frequency dependent. So in this case, we also looked at the microglia. And this is showing you dark flicker. We, in this case, did not find an increase in the microglia number. So it doesn't seem like microglia are being recruited to the area. But we do see an increase in cell body diameter and a decrease in process length, and also an increase in the percentage of microglia with amyloid beta. So we're finding that 40 hertz light flicker alters microglia morphology and results in higher colocalization with amyloid beta. All right, so then we wanted to know if this might actually have an effect on plaques. So I mentioned earlier, so far we've been looking at soluble amyloid beta in younger mice that don't have plaques yet. And that soluble amyloid beta is thought to be toxic, and we would like to be able to get rid of it early in the disease. But if you're also want to treat people who already have plaques, it would be nice to know if this would have an effect on plaques. So to do that, we're using 6-month-old 5XFAD mice. These have plaques, and we're defining plaques as amyloid beta deposits that are greater than 10 microns in diameter. And this is the way we treat them. So the previous studies I've shown you so far, we treated the animals for 1 hour, we sacked them an hour later, and that was it. So it was a relatively acute effect. We hypothesized that we would need a longer or more extended treatment to have an effect on plaque, because what I haven't shown you is that we know that the effect wears off. So if we look at it over several time points after that initial treatment, we know that it wears off around sometime between 12 and 24 hours after the initial treatment. So for these animals, we stimulated them with flicker for 1 hour per day for 7 days. So, and this is just showing you a staining where the amyloid plaques are these green circles here. This is showing you with dark, and this is with 40 hertz flicker. And what we find is that the average plaque number here is decreased with, and the average plaque core area is also decreased. And we don't fully understand exactly what's going on here, but one thing that could be going on is if we get rid of the soluble amyloid beta, some of the amyloid released from the plaques that then can get cleared out by the microglia. So we're finding that 40 hertz light flicker reduces plaque number and size. And then we also looked in some other mouse models. So what I've shown you so far was in 5XFAD mice. But we wanted to know if this was specific to this one type of mouse or if it would generalize. So we looked in aged wild-type mice as well. These animals have elevated amyloid because they're older. They're wild-type. Their amyloid beta is produced from its endogenous promoter. So that's a nice test of whether or not this has to do with the transgenic expression in the mouse models. And what we find is we can also reduce amyloid in these animals. The levels are pretty low, so we could only look at one of the amyloid proteins, not the other. And then we looked in another mouse model of Alzheimer's disease, the APP-PS1 mouse, and we find that we can also reduce amyloid in that mouse. These were all the 40 hertz light flicker experiments looking at visual cortex. Now, we also looked at another protein that's implicated in amyloid in Alzheimer's disease, and that's tau phosphorylation. And we looked in the tau P301S mouse model. We can reduce tau phosphorylation with 40 hertz flicker. We don't fully understand the effect at this point. All right, so what we're finding is that there are deficits in gamma oscillations during shortwave ripples. That gamma, as I mentioned, is associated with learning and memory deficits altered in Alzheimer's disease. These deficits occur early in the disease. And really interestingly, driving that gamma reduces pathogenic proteins. We can do it with optogenetics. We can do it with a non-invasive light flicker. And one of the effects of that manipulation is that microglia are recruited to the area, and they change their morphology, and we think they're engulfing amyloid beta. We can do this in sensory cortex anyway, non-invasively. And this is showing you a novel link between specific patterns of neural activity, this particular frequency, and the brain's immune system. The microglia are one of the primary brain cells that's implicated in the immune system. All right, so overall I've shown you that sharp-wave ripple reactivation predicts performance in a spatial navigation task. It's providing a novel link between place field codes and decision making. And sharp-wave ripples are lacking in an AD mouse model. There's fewer of them, there's less gamma, and this occurs early in the disease. And because in Alzheimer's disease, one of the earliest deficits is in spatial navigation, and because sharp-wave ripples have been implicated in spatial navigation, we think that this could be a part of the reason why behavior is going wrong in the disease. That's yet to be shown. And then we find that if we we can reduce the molecular pathology of the disease and increase microglia engulfment. And to me anyway, this really revises the way that I think about this plot here. So instead of just thinking, oh, proteins accumulate, they disrupt neural activity, and that leads to learning and memory deficits, there's really an active relationship here between that neural activity. It could, in fact, be feeding back and affecting this protein accumulation. All right, so going forward, we really want to figure out this relationship here. So how can we predict the disease from this neural activity? That would be potentially useful as a way to detect the disease early, monitor the disease, monitor treatments, as well as just simply to understand this relationship. And then we also need to figure out ways to drive gamma non-invasively, a way that works in the cortex, so that we could potentially disrupt this protein accumulation simply by driving neural activity. We also want to apply this to new diseases. So I've been collaborating with Michelle Pardue on looking at how this might affect the retina and retinal degeneration. And we also would love to be able to apply these to humans. So that's where we need some non-invasive ways to drive gamma and also to detect these deficits. All right, I'd like to thank my collaborators. I realize now I mentioned Hannah, who did all of the cellular molecular biology that I showed you at the end. She's a fantastic graduate student in Li Wei Sai's lab. But there was also a lot of help from other members of that lab. And the VR system that I showed you, I set up with Giovanni Tole Franzesi and had a number of wonderful undergraduate students who helped us get that up and running. And then the first part of my talk was all work done with Lauren Frank and in collaboration with other members of the Frank lab. And we've had some wonderful other collaborators. As I mentioned, Emery Brown gave us that great Flickr idea, and Dimitri and David helped us out with the VR system. And then I've had a wonderful collaboration with Craig Forrest here, and some others back at Boston and at Emory and the VA. And lastly, I'd like to thank my funding. All right, thank you very much.
Questions for Annabelle?
So you showed that you lost ecotoxin. Do you think you need excitatory?
We showed that GABAergic transmission is required for the effect, but is glutamatergic or excitatory transmission, yeah? I don't know. I would guess yes, because the current prevailing theory is that oh, yeah, go ahead.
So I was just curious, a lot of the lab analysis with the Flickr was from the sensory cortex, but did you look at the hippocampus to
Yeah.
actually see if there are any changes?
Yes, we definitely looked at the hippocampus. We can entrain the hippocampus, but the overall effect seems to be less than in visual cortex, perhaps not surprisingly. It actually surprised me that we can entrain hippocampus at all, to be honest. And we don't see an effect on amyloid, and we don't really see an effect on microglia. So we think that it's sort of having an effect on hippocampus, but it's simply not strong enough.
Do you think, I mean, from Williams' lab, right, have you tried like Chronos or anything like that to actually the infrared?
Oh, to do it non-invasively with optogenetics? We haven't tried that, but that's entirely possible that that would work. Yeah. Yeah, it's just that, I mean, it would work potentially in a mouse. The thing is, the tricky part about hitting the hippocampus, in a mouse, it's not that hard. In a human, it's hard. It's much smaller, and it's much deeper. So I don't know if it will work in a human. Go ahead.
So going back all the way to the decoding
Yeah.
side of things, a lot of the place cell work that you see has these very structured choice environments, and I'm curious, I know there's been some work in like more naturalistic, heterogeneous, complex environments. Is there any understanding of these ripples in a more diverse environment, what they might be encoding? I'm just wondering if there's more subtlety yet.
Yeah, so there actually isn't a lot of work where people are doing really complicated environments. the standard is like an open field or a linear track. But there is one study that comes to mind where they had a lot more reward locations. So it was an open field with a lot of reward locations, so there were a lot of different paths that you could look at. And they found that you could also predict the animal's path or the animal's ultimate location from the ripple activity. But did you mean something more like?
I was curious first if the ripples just existed, if you
Oh,
were
yeah.
exposed to much more, and if they were sort of coherent and sort of stayed, still had this predictive power.
Yes, they definitely have predictive power, and I think it's easier to see actually the more, because the more paths you have. It gets tricky though, because you need to do these predictions, you need to record a lot of neurons. And so we start running into sampling problems at some point. Oh, yeah, go ahead.
So can you go over the timeline a little bit? It sounded like you were saying that when you could stimulate fairly acutely and get changes in the amyloid beta very quickly, like within a few hours. Was that what you said?
That is true, yeah. So the timeline. The animals are exposed to flicker for an hour or optogenetic stimulation for an hour. They sit for an hour and then they get sacked. And you can see change in microglia and amyloid on that short time course.
And so does that surprise you? That kind of surprised me that you can see it so quickly. Like you'd actually get a change in the amyloid
Levels,
content,
yeah.
like levels?
Yeah, I guess it doesn't surprise me because, I mean, at that point, we've been thinking about amyloid dynamics for a while. So I think, people often think about plaques, and plaques stick around in a way that's different, fundamentally different than soluble amyloid. Soluble amyloid is being turned over. It's being produced and it's being cleared away. I don't anyway, and I don't think the field knows the exact time course of that process, but it is an ongoing process. So when we looked at the time course of our manipulation, we see that there's a decrease at 1 hour. It goes, I think, even lower at 4 hours. It starts to rebound around 12 hours, and then it's back to baseline at 24 hours. So there seems to be a real, potentially really interesting time course of amyloid clearance and amyloid production. I didn't show you, but we also see changes in what we think reflects changes in amyloid production. So that could be part of the time course that we're seeing as well.
And does the soluble amyloid turn into the plaques? Like, do we know that?
Well, there's definitely amyloid in the plaques. So the thinking is that the plaques, exactly why you get plaques and not just soluble amyloid is under current investigation, but the sort of the overall thinking is if you have enough soluble amyloid, they'll form into plaques somehow. So, but it also contains other proteins as well.
We thank our speaker one last time. We can maybe take some questions offline. In addition to her scientific prowess, Annabelle is also one of the co-organizers of the neurosocial. Did you want to make?
I forgot all about it. So there's a neurosocial on Friday at 5 at Park Tavern. It's kid-friendly. If you have children, you can bring them. It's on the park. They can play outside. So please join us.
All right. Let's thank our speaker.